would like to start off today by asking everybody a question. Have you ever wondered how a chameleon can almost instantaneously change its colors in nature? Or how, for example, an octopus is able to not only change its coloration, but also its pattern formation? These phenomena in nature happen thanks to something extraordinarily small, tiny microscopic crystals that can be found in this specialized cell that you see here called the iridophore. Now, within the iridophore, there are specialized compartments or membrane-bound organelles, termed the iridosome, where these crystals form. And the cell is able to very precisely control this crystal formation, from everything to the crystal's size, its shape, and even its chemical composition in ways that far exceed our man-made abilities in material science. Now, here are just a few examples of a variety of crystals that can be found in these cells. So some of you may be wondering, do we humans have these specialized cells? Do we have iridophores? And the answer is unfortunately no, we do not. When crystals appear in humans, they often signal disease, such as the case with kidney stones and gout. But in nature, organisms such as fish, reptiles, insects, and many other organisms, they're forming these crystals not as pathological deposits, but rather as functional biological materials. Now, these crystals are mostly made of guanine, the molecule that you can see here in the center. And to some of us, this may sound familiar, and it should, because guanine is the G that comes from both our DNA and RNA. So guanine is essentially already in the cell's molecular language, but iridophores are able to do something extraordinary. They're able to concentrate and organize this material, the molecules of guanine, to create functional crystals that are able to interact and reflect the light, and they serve a variety of purposes and functions in these organisms, from everything to color, to camouflage, and even vision. And these are essentially acting as biological optical tools that the animals can then exploit. So throughout my PhD, I've been trying to answer several questions, and one of the larger ones is, what molecular mechanisms do organisms use to actually control and regulate crystal formation? Now, in our lab, we're mostly using the zebrafish as a main model organism for several reasons. One being that it forms these guanine crystals, so it's wonderful. We can see them in a live system. It's also relatively easy to perform genetic manipulations on this model organism. As well as the larvae, their offspring are transparent, making them extremely accessible for us as researchers to perform different experiments, especially in the world of microscopy. Lastly, crystals form as early as 48 hours or 2 days post-fertilization, and so we can start from a relatively early time point to track the crystals and follow them throughout the development of the organism. Now, to answer the question of how these cells are making the crystals and to better understand that, we first started by looking at the proteome. This is a volcano plot where the proteins we found to be enriched are here on the right in purple, and the proteins that are down-regulated are here on the left in pink. And what became extremely apparent very early on is that these cells are very metabolically specialized. Now, what do I mean by that? When we look at different analyses, such as gene ontology enrichment analyses, or essentially the pathways that are being enriched, one thing that stood out is the purine biosynthetic process. But it's a bit more complicated than that, because we could also see simultaneously that while these processes are being upregulated, they are as well being downregulated. And so this was really interesting to us because it suggests some type of fine-tuning mechanism by the cell where it can exploit these processes and tune it exactly for its needs, in this case for crystal formation. And so what I'd like you to take away from this slide is that iridophores are metabolically specialized cells, and they have this significant enrichment in the de novo purine metabolomic pathway. Now, the next question that we wanted to ask is this metabolic specialization directed towards guanine? So it's not enough that we see this upregulation in purine biosynthesis. Actually, the family of molecules that make up purines, there are several of them. Guanine is one of them. But we wanted to know, is this specific for guanine? Because at the end of the day, the crystals that we're looking at are made from guanine. And so in order to do that, we mapped the biosynthetic pathway of guanine, where it starts here at the top. Maybe it's hard to see the laser, but with this molecule IMP. And what I'd like you to notice is that all of the proteins that subsequently follow and are the enzymes that are marked in purple were found to be enriched and upregulated in this system. And I think that you can appreciate that this is a rather complex pathway involving multiple steps and a variety of proteins that are needed to ultimately create our sink of guanine. So the answer is yes, iridophores do indeed reinforce the route to guanine. And as I mentioned before, this makes sense because our crystals are made from guanine. So we then wanted to take this a step further and to really understand if iridophores need to strongly increase guanine production to build crystals, what is supporting this metabolic demand? Because I told you in the beginning, this is a special ability of the cells. Not all cells can increase guanine to this amount. And this essentially puts some kind of stress on the system. So what do I mean by that? In order to create this upregulation and to really push forward guanine biosynthesis, the cell needs a few different things. One of them being ATP, or vast amounts of energy, as well as carbon and nitrogen sources, also to create things like the purine rings, different amino acids, as well as cofactors for essential enzymes in the pathway of the guanine biosynthesis. And so we really quickly were able to understand that guanine biosynthesis is not only a purine pathway problem, but it's rather a whole cell metabolic problem. And so we were really interested in understanding how the cell is able to deal with that. And when we looked at this list here of what I just described to you in terms of the biosynthetic needs, something really popped out, and that's the fact that a lot of these processes are connected to the mitochondria. And so we really started to wonder and hypothesize if the mitochondria in our system could be contributing or helping support the right metabolic environment for guanine biosynthesis, specifically for crystal formation. So in order to answer this question, we then went back to the proteomics, and now you're looking again at the upregulated proteins here in purple. But now we were able to find and identify proteins that were enriched and connected to the mitochondria. So mitochondria are indeed abundant in these cells. We can detect them, we can find them, and a lot of these proteins are actually connected to the electron transport chain, so cellular respiration. And once we knew that they're present, we then wanted to know where they physically located inside the cells. Here you're looking at an image that shows multiple iridophores. The mitochondria are marked in this pinkish purple color that you see here. And I think that you can appreciate not only are they abundant in the cells and they're quite packed full of mitochondria, the crystals are also in very close proximity and kind of running in parallel here along the crystals. So the mitochondria are in the right place at the right time. And we then wanted to ask, like any good biologist, what happens if we mess up the system? If we disrupt mitochondria and their function, what will essentially happen to crystal formation? And here I'm starting by showing you a panel of what our wild type look like. The mitochondria, again, are in this pink-purple color. We can see them running along our crystals. And here on the right, a beautiful image of an elongated guanine crystal. But when we started to look at mitochondrial dysfunction, and we did that in the context of a mutant, a known mutant called MPV17 in our field that models mitochondrial dysfunction, not only did we see the mitochondria themselves are no longer happy and functioning properly, they're fragmented, but we saw a huge shift in the crystal shape. If you compare these two images, the top and bottom at the right, you can see the one on the bottom is much more square, much more isotropic, and very different from what we get when the mitochondria are functioning properly. So for a long time, we've known that crystal shape is ultimately controlled by its composition. So the next thing we wanted to look at was the crystal's composition, and something that stuck out very quickly were the levels of hypoxanthine. In these mutants, there were much higher levels of hypoxanthine. Now, I didn't get to tell you a lot about this player, but now I'll share with you a few important insights. So I told you up until now that guanine makes up the majority of the crystals. The crystals are mostly made from guanine, but in reality, the crystals are not 100% guanine. They're actually solid solutions of another purine derivative here called hypoxanthine. And this is important because we established back in our previous work in 2024 that actually the amount of hypoxanthine directly impacts crystal growth and crystal formation. So hypoxanthine can inhibit certain facets during crystal growth, and the higher levels, if you look here on the right, the highest levels of hypoxanthine result in a crystal shape that looks much more square-like and much more isotropic. So this was very similar to what we were seeing in the case with the mitochondria. And so we now knew that mitochondria dysfunction is altering crystal shape, and it's also altering chemical composition. But what we didn't understand is, how is this happening? How is a change or shift or something that's not functioning correctly in the mitochondria ultimately resulting in this change in the crystal? And so in order to answer this mechanistically, we took a pharmacological approach to inhibit different complexes in the electron transport chain. This is an illustration of the electron transport chain. And unfortunately, because of time, I'm not able to go into all the experiments that we performed, but I would like you to focus on this first complex here on the left. This is the first complex of the ETC. And we used a drug called rotenone, and we got significant results that I'd like to share with you today. So when we treated these fish with this drug that blocks complex 1, we can see here in our wild type or our control at the top that we have lots of guanine crystals. They're very nicely elongated like we're used to seeing. But in our treated fish, not only did we have less crystals, we saw a significant shift also in the crystal shape. So again, very similar to what I showed you previously in the mitochondria mutant, where the crystals become much more square-like. And indeed, when we then quantified the amount of hypoxanthine that is present in this system, we were able to see, just like the mitochondrial mutant, treating with this drug rotenone also results in crystals that have much higher hypoxanthine. And so now I'd really like to connect this and put into perspective of what's happening in the system. I mentioned that we blocked complex 1. Now, blocking complex 1 here in the electron transport chain, it ultimately stops this molecule here, NADH, from donating its electrons and regenerating something called NAD, which is a cofactor in this system. And you don't have to just believe me, we can also quantify that, and this is a graph here showing you that the amount of NAD is reduced from the wild type to the mitochondrial mutant that I showed you before, as well as in our drug-treated system, which makes sense. But why is this important, and what is the connection to NAD? So I'd like to take you back for a second to the biosynthetic pathway of guanine, where at the top here, we have this enzyme, IMPDH1B, which is converting this first step of IMP to XMP, and this enzyme is heavily dependent on the amount of NAD. This is a cofactor that the enzyme needs for it to function properly. So without NAD, we essentially bottleneck our system here, and the pathway cannot move forward to create vast amounts of guanine like we're used to seeing. And rather, what happens is the pathway gets shunted and kind of shifts at the top here, where we're able to see more inosine and more hypoxanthine, which I show you here in graphs also on the left, both in our mitochondrial mutant model as well as in our drug-treated fish. And so ultimately, this is where the hypoxanthine is coming from and what's being incorporated into the crystals at the end of the day and resulting in this shift in morphology. So I think what's really interesting to me personally in this work is that our system, we're looking at mitochondria not only being the powerhouse of the cell and providing energy, but rather they're able to help and maintain the right metabolic conditions that are needed to make the right kind of crystal. And if I could just leave you with a few take-home messages from my talk today, I would like you to remember that in our system, at least, when the mitochondria are functioning properly, we have proper NAD availability. We also have high guanine-directed flux, as well as controlled inosine and hypoxanthine levels, which results in normal crystal growth, as you can see here. But when the mitochondria fail or they're dysfunctional, we have NAD being reduced, the levels are dropping, this purine balance starts to shift, and we get increased levels of inosine and hypoxanthine, as I showed you today. And this is essentially why the crystals become shorter due to the change in their composition. And we end up with crystals that look like this. So with that, I'd like to end my talk and my entire lab, the Gore lab, as well as my advisor, Dr. Dvir Gore. I've highlighted people's names here in pink who directly contributed to the work that I presented today. I would also like to thank the organizers, of course, and Limor and Edith for inviting me here to speak, and for you for listening. Thank you.